Good evening, everyone. Thank you for coming tonight, especially on this hot, steamy night, although it could be worse. It could have been last week, right? My name is Sharon Gross, and I am a senior science advisor in our ecosystems mission area, and I also am the program coordinator for invasive species here at USGS. Two high-profile species that we're going to talk about tonight are Asian carp in the Midwest and Burmese python in the Everglades. And hopefully you all had a chance to see some of our specimens here. I was going to make a bad joke about, well, we had a live one, but it's not there anymore. But I decided once the lights went down, that probably wasn't a good idea. All we have is the skin. But it gives you an idea of the breadth of some of the issues that we're facing, whether it's an invasive snakehead fish or Burmese python or zebra mussels. So these pythons and Asian carp, I think, have important lessons for us. Neither one of these were imported with the intent of being released in the wild, yet they were. So I think that's an important lesson that we get from all of this, is that sometimes things happen and you always have to take the necessary precautions to try to keep keep things in check. So we have two other speakers tonight, and I will give you a little bit more introduction on them, Bob Reed and Cynthia Kolar, and I'll give you an introduction on them right before they speak. So I've been with the USGS for about 10 years now, working in invasive species, and before that I was with the Fish and Wildlife Service working on their invasive species program. So I actually, when I was thinking about my intro today, I realized I have about 20 years working on invasive species. So they've been around quite a while, and our business does not seem to be diminishing. So what we're going to do is just give you a little overview of a couple of these species, the more high-profile ones, and talk about some of the research that we're doing that's providing some really valuable information. Now, the natural areas in the U.S. are under siege, I guess is a good way of putting it, from many invasive species. Some estimates indicate as many as 6,500 invasive species are now in the U.S. And there are some economists who have estimated the cost of these species to be upward of $137 billion annually. So there is quite a cost to not only other species, native species, but other native ecosystems and people as well. And the range of the impacts, it's a variety of species, as you can see here. Giant salvinia up here on the left are changing many lakes in the southeast. That looks like it is a solid mass, but there's actually a lake underneath of that. And just imagine if you had lakefront property and this is what you looked out at every day. Not a very pretty sight and not easy to boat on for sure. Nutria, and those really are orange teeth from South America, have been found in many wetlands in the United States. They've actually been almost eradicated in the Chesapeake Bay, but they eat wetland plants and have been, what they do is they eat the bottoms out and they change the marsh into mud flats. So they change it from a nice marshy area into an open water, and it changes the dynamics of the wetlands. This may be something that many of you remember from the news, probably, I guess it's been 10, 12 years now. The northern snakehead fish, we had our own population explosion, first in Crofton Lake out in Maryland, and then we found a population in the Potomac, which is still there. Once these become established, it's very difficult to get rid of them. We're not 100% sure. We haven't seen too many impacts from the snakehead, but the population has not exploded like we anticipated that it might. Salt cedar here, or tamarisk, is found in the southwest, and it is pushing out some of the native riparian plants. So it grows along rivers and streams in the southwest, and it really changes the dynamics along the stream so that you get these dense stands of vegetation that it's very difficult for people to get through and even animals to get through. And then here, some of you may remember this, kudzu, the plant that ate the sow. It was introduced for erosion control in the south and ended up just basically growing over everything. Some of the other species, we have one here on the table called the brown tree snake. It was introduced into Guam after World War II when military transports coming back from Indonesia and the Southeast Asia brought it into Guam, and it decimated the bird populations. Because there were no native snakes, the birds that were ground-dwelling birds on Guam had no mechanism to deal with predators such as snakes, and they basically have killed the majority of the bird populations on Guam. We are trying very hard to try to keep the snake from getting into Hawaii and some of the other Pacific islands right now. Feral hogs are a big problem in the southeast U.S. and other parts of the southern U.S., Mississippi, Alabama, and also in Hawaii, in national parks, in refuges, in other state-managed lands. They come in and they root up a lot of the natural habitat. So they disrupt the habitat for a lot of the other native species. So they become quite a problem, and many of the southern states have hunting programs to try to deal with them. Cheatgrass in the western United States is a grass that came from Southeast Asia and Africa, and you can see there it looks like kind of a savanna-type grass, and it is accustomed to growing very early in the season before the native grasses have an opportunity to become established, and it's also become a real problem for fires. In the western United States, this has become quite a fuel for fires, and it has created a fire cycle that is very unnatural, and the native plants have not adapted to this. So it's created kind of a cycle now where the native plants can't take hold. So we just keep having cheatgrass coming back again and again and again. Leafy spurge, which is in the northern prairie area, has taken over and pushed out many of the native grasses. It is poisonous for cattle, so the farmers don't like it, obviously, but as again, it outcompetes many of the native prairie grasses as it has changed those ecosystems considerably. And then, of course, there's zebra mussel, which kind of started the whole government the government really got involved with invasive species on the aquatic side with the invasion of zebra mussel in about 1990. And it has moved from the Great Lakes down the Mississippi and is now in the western United States as well. It coats the bottom of the waters and or the lakes and the streams where it's found, primarily the Great Lakes, but it also will grow on the bottom of boats, so it's a real problem for boaters and fishermen and other people who utilize the waters. So, I guess all the invasive species that I've shown you thus far are causing some pretty severe impacts, but not all non-native species cause impacts. Some of them will grow, some of them will linger, some of them will wait for years before they begin to cause problems. So it's very difficult as scientists for us to be able to predict which of these species will become major problems and which of them may be able to learn to coexist with our native species and not cause us a lot of issues. At the USGS, we have scientists across the U.S. who are studying these various invasive species. Again, trying to figure out which one of them will become problems, how we can prevent them from being introduced, and once they do become established, what we can do to try to prevent their spread or to control them. Our goal here is to learn about these species and their life history, what they eat, where they live, what temperatures they like, what temperatures they don't like, and this knowledge then helps us identify some of their weaknesses that we may be able to exploit to try to control them. We provide some of this critical information to other land resource managers, so national parks, the wildlife refuges, people who are responsible for trying to keep our natural system in check. So I guess just to give you a little background, the big question, how did they get here? How did these non-native species or these invasive species show up here to begin with? Well, there's a variety of ways. One way is the ballast water of ships. Zebra mussels are the classic example that come in the ballast water of ships. Ships leave another country, they fill up their ballast tanks with water, come across the ocean, come into our ports, push out the ballast water, and then load up with some type of a product. And, they move their ballast water around all the time to try to stabilize the loads on the ship. So ballast water spread organisms not only to here, but from here as well to other countries. They also come in agriculture. I don't think I have a picture here, but many of the plants like cheatgrass came in as seed in agriculture. Obviously, it wasn't what we intended, but they come in just as part of the seed stock. Plant cultivation is another way that organisms. This little plant called a purple loosestrife was actually brought in as a beautiful cultivated plant, but when it gets loose in the wetlands, it takes over and pushes out all the native plants. This was brought in for erosion control called beech vitex. Great idea at the time, but unfortunately now it's pushed out all the native dune grasses. So in many cases, things are brought in for a reason, and it seems like a good idea at the time, but there are many unintended consequences for these. Another pathway is through an aquarium trade. Here we have aquarium plants that are brought in, and then aquarium fish are often brought in. People raise them in their aquarium, but many times when they don't want them anymore, they'll turn them loose. This is a big problem in Florida because in Florida, if you turn something loose, there's a high likelihood that it's going to survive. So many of the native fish in Florida have been pushed out by aquarium fish that have been released into the wild. So globalization has really increased the ability for species to move all around the world, not just to the U.S., but also from the U.S. to other countries as well. And you can see here, the number of imports has increased exponentially, and we now have trade routes all over the world. And everywhere we have a trade route, we have a potential for spread. And I believe that this one talks about the shipping trade. It carries more than 90% of the world trade, and it's increased, it's tripled since 1970. So through that pathway, we have many more opportunities for things to be moved about and then potentially introduced. Now, one of the areas that USGS has done quite a bit of research is looking at fish species. So this slide shows you the number of fish species that have been introduced into the drainages. These little tiny things are called hydrologic unit codes, and those are drainages in the U.S. And you can see the green ones and the pink ones are where we've had more than 30, up to, I don't know, whatever that number is, I can't see what it is from here, introduced. But you can see in Florida and on the East Coast, we've had quite a few introductions, and again on the West Coast. Now, a lot of these were introduced for a variety of reasons. Some of them were sport fish back in the 40s, 50s, and 60s, and even back in the late 1800s. We introduced many species to different areas for sport fishing purposes. Others are introduced through aquarium trade or through other mechanisms. So this gives you just a general idea of what some of the origin of these foreign species are. This slide shows both fish and reptiles, but it also shows you that there are many commonalities in the U.S. with South America and then also with Asia. So we are, in many cases, have similar habitats, and these species are able to survive here. And this shows you some of the pathways how fish can be introduced into the U.S. And it also gives you an idea of the differences depending on whether you're in Montana or Florida. In Montana, obviously, fish from an aquarium trade, that's not a pathway that's not really a problem, but sport fish have been introduced pretty significantly into Montana. So the number of non-native fish in Montana are primarily through the sport fishing trade, or for sport fishing. Whereas in Florida, the majority of the non-native fish have come in through aquarium release. And, other areas here, bait release is a big issue in the Midwest, and also in places where you're doing a lot of sport fishing. So some of the research that USGS does, this is one of our scientists who's working on predicting where species may become established, and then utilizing some work with climate change models to predict what might happen as it gets warmer. And something like kudzu, we can see that we may have a difference depending on the climate change or on the warming caused by climate change in how fast or how far it might spread. And we do these kind of models for a variety of species, many plant species, but now we're also doing it for species like pythons and Asian carp to try to get and zebra mussels to try to get some kind of a prediction on where we might need to worry about these species next. For invasive aquatic invasive species, we have a database, non-indigenous aquatic species database, and it has not only distribution information and location information for many species, primarily aquatic, but there are also some reptiles and mammals with the database as well. And it also has, as part of this system, an alert system. So if you're with a state and you are trying to track the spread of snakeheads or the spread of Asian carp, you can sign up on our non-indigenous aquatic species alert system, and you can tell it knows where you are, and when a new location for that species is recorded, it will send you an alert. So it allows you, if you're a manager in a state, to be able to track where that species is spreading, to give you just a bit of a heads up when you might need to worry about it. Some of the other research that our scientists are working on is looking at the interaction of native species with some of these introduced species. This one has to do with the salt cedar or the tamarisk, the plant species I told you in the Southwest. We're looking at interactions with a Southwest willow flycatcher, which is an endangered species there, which has begun to utilize the tamarisk for habitat. So sometimes, even though the non-native species has we would expect it to have a negative impact, some species will use it as well, so that it can have, I don't want to say a positive impact, but it can have a beneficial use as well. And then these are some of the other species that potentially use it. So I just wanted to give you just a general overview. And now we're going to have Cindy Kolar to come up and to our next discussion on Asian carp. She is our USGS Science Authority on Asian carp and is a world authority. She basically wrote the book on Asian carp. So we'll let her talk to you a little bit about it.
Boy. Thank you, Sharon. So I'm not sure how many people around here have heard about Asian carp, because it's not really a problem in this region of the country. Asian carp are a big problem throughout the Midwest, in the larger river systems in the middle of our country. And Asian carp really encompasses 4 species, 2 of which, like in the media, when you hear about Asian carp here, it's pretty much 2 species, bighead and silver carp. This one over here is a bighead. Both species get very big. Bighead can get up to 100 pounds or more. And silver carp, I think a maximum is more like 40 or 60 pounds. But they're great big fish, and they're planktivores. So they filter plankton, planktonic plants and animals out of the water. And it is silver carp that jump. So I've got some pictures later where they explode from the water. They jump high out of the water. And it can be like 14, 15-pound fish that are jumping and have hit people. If you're out, our scientists that are out there collecting fish or just working in the area, they have cages around them, like Dwayne Chapman, who's one of our scientists here, has a cage around himself to protect. And especially like the throttle over here, so fish don't hit it. So these fish were introduced in the 1970s, and they were brought in for specific purposes, but they quickly escaped those purposes and got into natural waterways. So where are they from? They're from Asia. Their native range, both species, is in China. And further north, this is silver carp. They get into Russia, and bighead carp is a little bit more of a southern sort of species. But they were brought over here on purpose in the 1970s for aquaculture, for human consumption. These fish they're tasty. They're not like the common carp that we're used to having here that are bottom feeders, and they can taste muddy or that sort of thing. These fish, they're actually very good. They're flaky, and because they eat at a different part of the ecosystem. They were also brought in for treatment of wastewater. And so this is 1970s, and people wanted to be greener. And so instead of having chemicals and that sort of thing, they wanted to bring fish in to help clean up municipal wastewater treatment, like in tertiary ponds. These fish are not only just in the US, they've been introduced on purpose widely throughout the planet. Over 100 countries have introduced either bighead or silver carp or both. And they were started being promoted starting like in the 1970s. And this has to do with improvements in ability to artificially spawn fish. So before you had to go out and collect little ones and bring them back and raise them, and now they were able to induce spawning. And mostly throughout the world, they've been introduced for human consumption. So it's for aquaculture, but a variety of other uses as well. So in the United States, they were first found in Missouri River and the Illinois River, and they've kind of started radiating from there. And so this one over here is for silver carp, and bighead carp over here. The darker colors are just where individuals have been collected. And they're thought to be reproducing in about 20 different states. These maps, Sharon just talked about the NAS database. You can create these maps real time, and here's the website right here. So I grabbed these today. And so if, yesterday the new state was reported, well, then we got the latest data here. So what's the problem? Well, they become very abundant. When they start spreading, they become the most abundant fish, and it's the biomass. If you take all the fish and you weigh them all, almost all of the biomass, over 90% sometimes, is that of Asian carp. And here we've got these are commercial fishers who are hoping to get some native fish, and they ended up with quite a bit of Asian carp there. And these are the silver carp I was telling you about that they jump out of the water. So this is in response to, they do this in response to boat motors or electricity. When we go out to electroshock fish for collection purposes, they'll leap out like this. They also leap, just kind of randomly, but then also below dams. So these fish were spreading. They started spreading up the Illinois River, and they were getting closer and closer to Chicago and the Great Lakes. And the Great Lakes provide about $7.5 billion of revenue to the Great Lakes states for commercial and recreational fishing each year. And so people became very concerned that these fish are going to get into Lake Michigan and then spread from there. And the only thing keeping them out, besides maybe they wouldn't want to move there, is that there's an electrical barrier that was installed around Chicago to keep invasive fishes from moving from the river into the lake. And then, so this is a growing concern. Everybody was getting concerned. And then in late 2010, there was some eDNA was detected, and that kind of changed everything. So eDNA is environmental DNA, and that is DNA that is shed, in this case, this is a bighead carp, is just shed into the water. And it's present in the environment. And so it can be shed through mucus or scales or urine or feces. So it's just, we are all shedding DNA, it's around. And so the idea is you can go out and sample water and you can work it up in the lab, find the DNA, and then go back and identify which species it belonged to determine presence or absence. And so in late 2010, there were some positive hits for eDNA for Asian carp, for bighead carp in and around Chicago. And so that got everyone quite excited, and there was a special committee that was put together that first comprised of federal partners and then also included the states. And a lot of monitoring started happening. These are just some results of some eDNA here, just to show you that it just got us all active in making sure that Asian carp do not become established in the Great Lakes. So USGS has been conducting research on Asian carp for 13 years or something like that, but it was all in the rivers, the big rivers in the middle of the country. So with the eDNA and this new focus on the Great Lakes, we started a variety of research to address the potential, the threat to the Great Lakes. We have some projects that are looking at, can they get to the Great Lakes? Can we detect them to see, in addition to eDNA, or could we improve it? Can they become established? Do the Great Lakes provide all the things that they need to complete their life history? Would they affect our fish populations? And can we control them? So we have projects ongoing in all of these. And the purpose for all of this research is not just because we're kind of curious. It's because we want to provide tools to managers. So when we want to be able, when we're looking to see can they become established, that's important to know. And where could they become established? Could it only be in particular bays? So we know that those are where we need to focus monitoring efforts. But for tonight, I'm just going to talk about these, give some examples of research we have ongoing in two areas. First, can we detect them? And then I'll talk about can they be controlled? So we have two projects looking at can we detect them. So I mentioned this eDNA, there's already an established method, and monitoring is going on every 2 weeks throughout the growing season of the year. And that's being conducted right now by the Army Corps of Engineers. That's going to be switching to the Fish and Wildlife Service soon. So the research that we have ongoing at USGS is looking at can we improve the method that's being used to detect eDNA in the water? And what exactly does a positive hit mean? Does it mean that there was a fish? Could it have been excrement from fish-eating birds? Could it have been shed off of a net? Or we need to validate the method to understand exactly what it means when you have a positive hit. And then we're also looking to see, can we improve the method, making sure the markers, the genetic markers that are being used are effective, and see if we can make the processing go faster. Our second project on can we detect them is, this whole detection thing is not a very glamorous sort of project. So we had bird poop before, and now this is actually looking at microbes that might be living in the gut of the Asian carp. So it's not only, Asian carp would not only shed their DNA, but they would shed through feces and urine, well, through feces, the DNA of the microbes that are the microbe flora of their gut. And a lot of times that flora is species specific. So we have a project going on to look at the DNA of the microbes that are found in the guts of silver and bighead carp. And the idea is that this would supplement and complement the work of the eDNA to help verify the method. And this is, it's actually very exciting. Oh boy. Okay. So then I'm going to move on to can they be controlled? There's a lot of different ways that you go about controlling an animal that you don't want around. something called biocides. These are chemicals that are used to control nuisance species. There's an extensive sea lamprey control program in the Great Lakes based on, well, they use a variety of methods, but mostly on chemicals. You can have biological controls, and the sort of thing that we're looking at had to do with pheromones and food cues. And then you can have physical control, where you either contain the species, to physically not let them somewhere, or you can control them by physically removing them, capturing them in nets or something like that. And we have a project on seismic energy that I'll be mentioning briefly here. Okay, so biocides. So biocides have been used to kill fish for quite a while. Maybe you've heard of rotenone. Another one is antimycin. So these are chemicals that are applied to the water by management agencies to remove nuisance fish, to restore a fishery that may be for recreational use. The problem with either rotenone or antimycin is that it kills everything. Because the way it works, you apply it to all of the water, you have to develop the concentration that will kill things in the entire water column. So it's not specific. So what we're doing is to see if we can develop a biocide that will target just Asian carp. And this sort of work, it was done quite a while ago in the 60s for sea lamprey, but there has been no advancement in the subfield of aquatic biology here. And so this is cool because we're moving things forward in things that have been stagnant for a while. And the idea for this came from one of our scientists who saw a talk where they were actually trying to help the fish. And so this is for salmon that are used in aquaculture that you buy in the grocery store, that farmed salmon, and they need to vaccinate them. And so there's a company that developed a technology where you can incorporate the vaccine, can incorporate the medicine into this micro matrix, into a little tiny particle. The fish eats it, and it's not until the gut where certain enzyme action releases the vaccine, because that's where it needs to be released so the fish can take it up and, not get sick. And so, if you can help something selectively, you can probably turn it on its head and use it to kill something selectively. So what we're doing is developing a molecule that has a poison. We need to figure out how much of that poison the fish needs to eat to die. But then we also have incorporated a trigger, something specific about the physiology and the biology and the digestion of that fish that will cause that toxin to be released. So the first thing we had to do, these are filter feeding fish. They bring particles in through their gills. So it's like, well, what size of particles do they eat? Because we can perhaps gain some selectivity just by having the right size of particle. So this is just from the literature. We have big-headed and silver carp here at the bottom, and these are the size ranges of particles eaten by some other aquatic taxa. Paddlefish, big-mouth buffalo, and gizzard shad are native filter-feeding fish. So we really don't want to hit these. And from the literature, we see that bighead and silver carp, kind of like this range right here, between 50 and 100 microns. And the only native fish is gizzard shad that kind of falls into that. There's a lot of overlap. Gizzard shad would get hit if that's the only way that we were able to select for bighead and silver carp. So that was just literature. And so we went through and did this in the laboratory to make sure that bighead and silver carp would be removing particles in that size range. And they did. These lines at the top were at the beginning of the trial, where you've got all the different size range particles in the water. And after 24 hours, they had filtered out these dashed lines. They filtered out a bunch of the particles, and it tended to be in about the range that the literature said. We can see this is a gut that's full of those particles over there. Okay, so we know that we can get the fish to eat a certain size particle. So that's like half the battle right there. Now we've got to figure out a way to release it, some kind of trigger that's specific just to bighead and silver carp. So they looked at the guts, they analyzed them all, take a look at the enzymes that are produced, and here's the different enzymes that are down here. And then the scientists figured out, well, which ones do we have the most difference between Asian carp and well, especially gizzard shad, because gizzard shad is the one where we had a lot of size overlap. And there's a digestive enzyme called trypsin. And here you see a big difference between the amount of trypsin in the guts throughout months of the year between silver carp and gizzard shad. So there's lots of trypsin in silver carp, but hardly any in gizzard shad. So we think that we can use trypsin as that trigger to release the toxicity. So what we're doing right now is coming up with a final design for this particle. We're working with a private company to have that produced. We're actually having the toxin produced, and then we're having the particle produced. And we're expecting to get the batches in the next couple months, and they'll be running trials in the lab with just bighead and silver carp, and then in tanks with the native species as well, to see if we can get some separation there. And if that works perfectly, then we'll do the same sort of thing, move up to ponds and see if we can get control, selective control in ponds. If it needs, there's a chance we'll have to rethink things, and we'll be doing the lab trials again. Okay, so I'm just going to mention briefly two other kinds of control research that we have on control for Asian carp. I said the first one was chemicals, biocides. The second major category is our biological controls. There's a variety of biological controls. What we're focusing on right here are attractants. So are there ways where we can bring Asian carp to a certain area? And the reason we'd want to do that is because we have some other tool to get rid of them. So it might be to bring them into an area to net them out, to get them into an area to treat with chemical. But if you can concentrate these fish, they're very difficult. These fish are very hard to find. They're very sensitive to vibrations. They're pretty wily fish. They're interesting fish. So we have two projects going on looking at attracting Asian carp. One is our sex pheromones. So females release sex pheromones to bring the males in for spawning. And you can induce, you can get fish to spawn artificially. And the way you do that is to inject them with hormone. So you inject the female with hormones, their ovaries start ripening, and what happens is the females release a pheromone that brings the males in. So a pheromone is just a chemical that says something to members of your same species. So in this project, they're inducing females, injecting the females with the hormone and caging them. And then they start releasing the pheromones and the males come in. And so they're doing some pond experiments this year to quantify the attractive effect. And the second thing we're doing is looking at food cues. So these fish filter algae and zooplankton out of the water. And so if you add a bunch of algae, they come, because they're excited, they want to eat it up. And so there's this idea of this algal seltzer, where these are little algal seltzers that have a lot of algae in them. So we can have some time release of algae. And so those experiments are ongoing as well. The last thing we have ongoing are some experiments looking at physical control. And in this case, we're looking at removing fish from areas, repelling them to contain them. And this is using water guns. That's what these things are right here. They're guns that you shoot underwater, and it kind of just shoots air. It's just air in the piston, and it makes a big noise in the water. Fish hate it, and these fish hate it probably more than others because they're very sensitive. And so the idea is where you have an area you don't want the fish, you'd be shooting these guns to keep the fish away. So right now we're looking to see what size of gun. We've done some preliminary work, and it works fine. If the fish are really close, they get killed. If they're further away, they scatter. And so what we're doing now is to find the ideal size of the gun and how often you need to shoot it, and so we can make it efficient and practical. We have a lot of other research going on Asian carp. I've just highlighted some of the neater things. But with all of this, we're not just doing this for research sake. We want to take each project and get it in the hand of managers. So we're working with the managers to make sure that we're providing the information that's going to be helpful to them in a way that is helpful, so that all this can be used in the field to protect our resources. And this is my name right here with my email. And if you want to know about Asian carp, there's a website, Asian Carp US, and that is like a clearinghouse for all of the work that federal and state agencies are doing right now. So thank you.
Thanks, Cindy. Okay, our final speaker tonight is Bob Reed. He is a research wildlife biologist, and he is a member of the boa and python specialist group with the International Union for the Conservation of Nature. Bob also got an award. It was a couple years ago. It's the Secretary of the Interior has a very prestigious award called the Partners for Conservation, and this was with his work with the Burmese pythons. I don't know if you saw the square risk assessment outside, but Bob's work with that, he was awarded that special award. Thanks, Bob.
Thanks, Sharon. Unless you've been living under a rock for the last few years, you've probably heard about pythons in Florida. But if you've been getting information from the media, you've probably been getting a lot of sensationalism, probably a lot of misinformation. With pythons, we're a few steps back from what Cindy talked about. We're still at the stage of trying to understand their basic biology so that we can get an idea of vulnerabilities that we could then use for control purposes. But here's a female python. She's sitting on her eggs, and pythons are clearly established and breeding in southern Florida. So how the heck did they get here? This is a snake that's from Southeast Asia, makes it from Malaysia to China, all the way over into India and Nepal in its native range. Well, the proximate hypotheses for how the snakes were released, there's a few of them. And these are how it got from captivity in the US out into the wild. So you've got things like intentional releases. We know that sometimes if a snake gets too big for someone to handle, they dump it hoping it'll find a little happy home outside. There are also unintentional escapes from captivity. These are big, strong snakes. They can escape from cages that aren't very strongly built. And then there's the idea of mass releases. The most commonly cited of these is Hurricane Andrew in 1992. We know that Hurricane Andrew knocked over some big animal importer businesses that were close to Everglades National Park. These are all the proximate hypotheses. I tend to think of them as more or less distractions because we know the ultimate answer. The ultimate answer is that we brought them into the country intentionally for the live animal trade. And this is the year from 1977 to 2007. And then we've got the number of pythons imported per year. And over a 30-year period, we brought in about 300,000 individuals of just this one species. With that kind of constant pressure, it's a near inevitability that some of them will make it out of captivity. And if they make it into an environment where they are capable of surviving, then there's a pretty good chance you're going to end up with a population. So we're going to go through some of the things that most people ask about these critters. How big do they get? Well, in Florida anyway, we've gotten them to over 5.5 meters and over 75 kilograms. That's about 17 feet or so and roughly my body weight. Although the average is smaller, about 10 feet long and 20 to 25 pounds. This is a 16.5-foot female that was taken out of the Everglades in April of this year. This is her head. The guy holding it is actually this guy. He's 6'6". He's got an enormous hand. So that's a big snake. There's no doubt that these are truly giant snakes as snakes go. This is just another way of looking at body size. We've got snout vent length down here. That's the length of the snake minus its tail. It's a standard measurement of length in snakes. And then on this axis, we've got its mass in kilograms. Multiply that by 2.2 if you want to convert that to pounds. You can see that they come out pretty small. They weigh about 1/4 pound when they hatch. And they're only maybe 20 inches long. But they grow very quickly. Not only do they grow in length, they start putting on mass. And you can see that these red dots are clustered near the bottom. Those are the males. All the truly big snakes are females. It makes sense to be a big female because you can produce more eggs. It doesn't really make sense to be a giant male because then you got to find big things to eat. That's hard to do. So you might as well stay medium-sized if you're a male. How many are there? Well, that's a tough one. We don't know how many Burmese pythons there are in Florida. And that's largely because if you want to estimate population size, what's usually done is to capture a sample of animals, mark them, and then release them. And then you go back later, capture another sample, and figure out the proportion of the ones you got that are marked. If you do that enough times, you can use those proportions to figure out how many are likely to be out there. In the case of Burmese pythons, no one wants to be releasing them. And so we don't have any good, credible estimates of population size. However, we're zeroing in on 2,000 pythons that have been taken out of the Everglades and vicinity. And most of those, about 90%, have been taken out in the last 6 or 7 years. If you ask snake ecologists, you're going to get estimates starting at about 10,000 individuals. We're going to go into why that is a little later. Okay, so why do we care? Well, the big impact is likely to be predation on native species. This is a very large snake, very unlike anything we have that's a native. Native species might not be used to this sort of a predator. The thing is, though, you almost never see them in the act. Snakes are very secretive. On the right here, we've got 2 of the 3 pictures I know of that show Burmese pythons actually eating prey items in the field. This is a black vulture that's being constricted here, and then this is a white ibis with a really bad camera phone picture. So you don't often see them eating things. That could lull us into a false sense of security. Here's a snake, about an 8-footer, not particularly big. This is on Key Largo. And this researcher with her hands clenched in fear is, she works on Key Largo wood rats. It's a federally endangered species. She was radio tracking one of her Key Largo wood rats that had a collar on it, and it led her to this snake. When they opened up the snake, they found her wood rat with the radio collar on it, as well as another one. At the time, that was about 3% of the total population of Key Largo wood rats in the world. So this is the very first snake found on Key Largo, and it had already eaten a couple individuals out of this very small population. Here's a list of the animals I know of that have been taken out of Burmese pythons. So things that were found in their guts. It's notable for a couple things. First of all, just the range of species. We're talking a whole lot of warm-blooded animals, basically any warm-blooded animal that they can fit in their mouth, as well as a few things that are not warm-blooded animals, like American alligators. The other thing you notice here is the size range. We've got things from house wrens, which weigh about an ounce, actually about 3/4 of an ounce, all the way up to white-tailed deer, alligators to 7 feet long. It's a really broad diversity and size range of potential prey that they're eating. As a snake ecologist, it's not just those individual records that impress me. This is about 20 pounds, actually over 20 pounds of body fat taken from one medium-sized female python. They're doing very well. They're in excellent body condition in general, much different from the boa constrictors, say, that I've studied in the neotropics. These animals are fat. So they're not only getting individual animals, they're getting enough of them to have a lot of energetic reserves. They can turn those reserves into growth or babies. So now I'm going to go through a few slides from a paper that we published a couple months ago. Down here we've got the year from 95 to 2010. And then here's the number of pythons removed per year from the Everglades and vicinity. And you can see that pythons were first recognized as being established around 2000. That's when the first scientists said, I think it's not just individuals we're seeing here. I think we've got reproduction. And then more and more started being collected over the ensuing years. We pulled together some historical data that included the ranger reports in Everglades National Park in the 1990s. The rangers would drive the main park road every day and record every road-killed animal they saw. We also had 1 year of nocturnal road surveys, where a park scientist drove the main park road at night, all the way down, all the way back, and wrote down every mammal that he saw on the roads. We then repeated this and expanded it quite a bit between 2003 and 2010 with about 55,000 kilometers of driving. We did that on the main park road. We did it in areas where the python had only recently arrived, and we did it in areas without pythons. So if you look back at 1996, and look at some of the more common species that we're seeing on the road. A lot of raccoons, a lot of opossums, and then smaller numbers of other critters. We repeated that between 2003 and 2010, and you see these little tiny bars? That's what we found. We got a total of 6 raccoons. There were many nights in 1996 where they got more than that in 10 miles. We got 5 opossums. We got no marsh rabbits. These declines come out to around 99% for some of these species. And then if you look at the areas where the pythons have only recently arrived, we get intermediate values for some of these. So not as many as the historical numbers, but more than in the heart of the python-occupied range. And then if you look in the areas that don't have any pythons at all, we're back to really high numbers. So we seem to have 2 patterns here. One is temporal. Things have changed over time. We've got fewer mammals than we used to. And we have a spatial pattern where areas with a lot of pythons, not many mammals, going out to areas with no pythons and lots of mammals. This is still correlative. It's not a causative relationship here, but those 2 lines of evidence suggest that pythons have probably knocked back mammals quite a bit in Everglades National Park. Central factor, perhaps the most important thing about snakes in general and pythons in particular, is detection probability. What is your odds of going out and finding a snake? And generally speaking, it's not real good. Probably 95% of the pythons that are captured in South Florida come from levees in the wintertime when they come out of these canals and bask along the levees, or they're from snakes that are found crossing the road at night in the summertime. The problem is that levees and roads comprise probably less than 1% of the total area occupied by pythons. This is a picture I took on an occasion when I was lucky enough to get a helicopter ride over the Everglades. And that's when it was impressed on me the vastness of this ecosystem. It's a huge, huge area. With that in mind, are we detecting 1 in 10 of the pythons out there? Is it 1 in 100? Is it 1 in 1,000? I'm pretty darn sure it's not this number. And whether it's this or this, either of those answers is fairly troubling from a control and management perspective. And detection probability even influences our opinion of where the heck are the pythons. So each one of these dots, these push pins, is a place where a python's been taken out of Florida. Now, based on what I know of the population, I'm pretty comfortable saying that they're established over this portion of the state. However, we've then got this string of observations that go north of there, and a lot of these are in pretty remote natural areas. So some folks might say the population goes up to here. So we're talking 4,000 to 8,000 square kilometers occupied by pythons, depending on where you think they are based on these maps. I think a lot of these are probably escaped or released individual pets, but I'm not completely sure of that. And then what do you do with an observation like this one? This is a single massive female that was found north of Lake Okeechobee in a fairly natural area. It just happened to be near this remote veterinary clinic where they were walking around outside and they found it. We know that pythons are amazing swimmers. We've documented movements of over 50 kilometers by individual pythons over the course of a couple months. If the population is here, and this female managed to swim across Lake Okeechobee, then yeah, that could be a disperser from the population rather than an escaped pet. But the fact that we've got a massive female, obviously in good reproductive condition, do we really care that much about whether she's part of the population or whether she's a released pet? We've got a lot of tough issues both from a scientific and a management perspective here. Okay, can we get rid of them? When I ask if we have tools for eradication, I mean the ability to get rid of every last python in a population. We can use visual searching and get some. This is actually a boa constrictor, which is introduced to Puerto Rico. We can use traps. This is a trap that we have on Key Largo that caught a python. We're developing eDNA technology that Cindy talked about just for determining presence-absence. But the short answer is, right now, we have no tool that allows us to really knock out every individual in a population. We're probably nibbling away at the edges. With that in mind, just talk real briefly about the northern African python. This is about as closely related to the Burmese python as you can get. And based on a couple observations on the west side of Miami a couple years ago, we did an intensive search in early 2010. And we started finding northern African pythons. And they're pretty big snakes. And we've established that reproduction is occurring. We've found pregnant females. We've found old nests with hatched eggs. We've found recent hatchlings. On the bright side, all these locations of pythons are in a very small area. In a case like this, with enough search effort, and even if they have low detection probabilities, I'm optimistic that we can get in there and get every single one. But it's only in these cases when it hasn't spread across a really large geographic area that we have that kind of hope. And with that in mind, what should we be focusing on? Well, the things that I keep harping on with folks are prevention, early detection, and rapid response. First of all, do whatever we can via education, regulation, etc., to prevent the animals from getting out in the wild. And then, if something new is spotted, try to make sure that you're watching for it and then respond. This is a yellow anaconda, a green anaconda, a reticulated python, a carpet python. All of these animals were observed by citizens. They were reported. There was a response to get that animal. Every single one of them was caught. This is incredibly hopeful. If we can continue that kind of thing, then perhaps we can prevent the next Burmese python invasion.